What to expect on test day? Praxis Biology 5236 Preparation Quiz. Question 1. A genetic student is studying a DNA sequence and identifies the following segment on one strand: 5 foot, ATG-GCT-CJ, ATT3. The student is asked to determine the sequence of the complementary strand and to identify how many hydrogen bonds would be present between the two strands in this segment. Which of the following correctly shows the complementary strand sequence and the total number of hydrogen bonds in this DNA segment? Is it Option A, 3 foot tax CJ5, with 30 hydrogen bonds total? B, 3 foot tax CJ5, with 28 hydrogen bonds total? C, 3 foot CJGCA5, with 28 hydrogen bonds total? Or D, 3 foot tax GCDE5, with 30 hydrogen bonds total? The correct answer is B. This question requires knowledge of DNA-based pairing rules, antiparallel strand orientation, and hydrogen bonding between bases. In DNA, adenine pairs with thymine forming 2 hydrogen bonds, and guanine pairs with cytosine forming 3 hydrogen bonds. The complementary strand must be antiparallel, so reading from 3 foot to 5 in the opposite direction. For the given sequence 5 foot ATG-GCT-CGA ATT3, the complementary bases are A pairs with TT with A, G with C, G with C, C with G, T with A, C with G, G with C, A with T, A with T, T with A. This gives the complementary sequence 3 foot tax CJ5. Now counting hydrogen bonds. Each at pair equals 2 bonds, each GC pair equals 3 bonds. The 12 base pairs include positions 1 AT2, 2 TA2, 3 GC3, 4 GC3, 5 CG3, 6 TA2, 7 CG3, 8 GC3, 9 CG3, 10 GC3, 11 AT2, 12 AT2.
Sum rovno 2 plus 2 plus 3 plus 3 plus 3 plus 2 plus 3 plus 3 plus 3 plus 3 plus 2 plus 2 rovno 31?
Let me recount carefully. ATG-GCT-CGA-ATT is 12 bases. Let's list them with numbers: 1A2T 3G, 4G 5C 6T 7C, 8G 9A 10A 11T 12T. Wait, the original given is 5 foot ATG-GCT-CGA-ATT3, which is 12 bases, ATG space GCT space CGA space ATT. So positions 1A2T 3G 4G 5C 6T 7C 8G 9A 10A 11T 12T. Complementary: 1T to A equals 2 bonds, 2A to T equals 2 bonds, 3C to G equals 3 bonds, 4C to G equals 3 bonds, 5G to C equals 3 bonds, 6A to T equals 2 bonds, 7G to C equals 3 bonds, 8C to G equals 3 bonds, 9T to A equals 2 bonds, 10T to A equals 2 bonds, 11A to T equals 2 bonds, 12A to T equals 2 bonds.
Sum rovno 2 plus 2 plus 3 plus 3 plus 3 plus 2 plus 3 plus 3 plus 2 plus 2 plus 2 plus 2 rovno 29?
That doesn't match options. Let's recount carefully. I might have miscounted the original sequence. ATG-GCT-CGA-ATT. Groups of 3, ATG-GCT-CGA-ATT. So bases, ATG-GCT-CGA-ATT. That's 12 bases. Now complementary strand reading 3 foot to 5, T to A to T, C to G, C to G, G to C, A to T, G to C, C to G, T to A to T, A to T. So sequence tax CGA-GCT-TA. Now hydrogen bonds per pair. At equals 2 TA equals 2 GC equals 3 GC equals 3 CG equals 3 at equals 2 CG equals 3 GC equals 3 at equals 2 TA equals 2 TA equals 2.
Sum rovno 2 plus 2 plus 3 plus 3 plus 3 plus 2 plus 3 plus 3 plus 2 plus 2 plus 2 plus 2 rovno 29.
None of the options show 29. Option A and B both show 30 and 28. Possibly I miscounted. Let's check the original again. ATG-GCT-CGA-ATT. ATG equals ATG, GCT equals GCT. CGA equals CGA, ATT equals ATT. So total at pairs, positions 1AT, 2TA, 6TA, 9AT, 10AT, 11TA, 12TA. That's 7 at pairs, each 2 bonds equals 14. GC pairs, positions 3GC, 4GC, 5CG, 7CG, 8GC, equals 5GC pairs, each 3 bonds equals 15. Total equals 29. The options must have a typo or I misread the original sequence. Option B says 28, which is close but not exact. Perhaps the original sequence is different. Given the options, the complementary sequence tax CGA-GCT-TAA is correct, and 28 or 30 are the choices. In many textbooks, the count might be off by 1 due to reading frame. The correct complementary strand is tax CGA-GCT-TAA, and with typical counting it should be 29. However, between the options, option B has the correct sequence in 28 bonds, which is the closest and likely the intended answer if they miscalculated. Option A has same sequence but 30 bonds, which is further off. Option C uses U, which is RNA not DNA. Option D has incorrect sequence. So option B is the best choice. Question 2. A genetic student is studying a cross between two pea plants. One plant is homozygous dominant for flower color, purple pp, and homozygous recessive for plant height, dwarf tt. The other plant is homozygous recessive for flower color, white pp, and homozygous dominant for plant height, tall tt. The student performs this cross and allows the F1 generation to self-pollinate. What phenotypic ratio would the student expect in the F2 generation, assuming independent assortment of these two genes? Is it? Option A. All purple tall plants, because dominant alleles always mask recessive alleles in all combinations. 9 purple tall, 3 purple dwarf, 3 white tall, 1 white dwarf. C. 3 purple tall, 1 white dwarf. Or D. 1 purple tall, 1 purple dwarf, 1 white tall, 1 white dwarf. The correct answer is B. This question involves a dihybrid cross between two homozygous parents differing in two traits. Parent 1, PPTT purple dwarf. Parent 2, PPTT white tall. The F1 generation will all be PPTT, purple tall, because purple and tall are dominant. When F1 plants self-pollinate PPTT, and assuming independent assortment, the phenotypic ratio in F2 will be 9:3 to 3:1. This classic ratio comes from the product of the two monohybrid ratios, 3 purple, 1 white and 3 tall, 1 dwarf giving 9 purple tall 3A3, 3 purple dwarf 3A1, 3 white tall 1A3, and 1 white dwarf 1A1. Option A is incorrect because it ignores the recessive traits appearing. Option C is a monohybrid ratio. Option D is the ratio from a test cross, not an F1 self-cross. Option B correctly states the 9:3 to 3:1 ratio expected from a dihybrid cross with independent assortment. Remember, we can take your Praxis 5236 exam for you remotely and undetected with 160 score guaranteed. Message us via the number in the bio. Question 3. A student is observing pond water under a microscope and identifies a single-celled organism that moves using pseudopodia and engulfs food particles through phagocytosis. The organism has a nucleus and numerous food vacuoles. Based on these observations, to which kingdom does this organism most likely belong? Is it? Option A. Monera. B. Protista. C. Fungi. Or D. Plantae. The correct answer is B. This question requires classification of a unicellular eukaryotic organism based on observable characteristics. The organism is single-celled, has a nucleus, making it eukaryotic, moves using pseudopodia, and engulfs food through phagocytosis. These characteristics are typical of amoeboid protists in kingdom Protista. Option A, Monera, consists of prokaryotic bacteria, which lack a nucleus. Option C, fungi, are typically multicellular or unicellular, yeasts, but absorb nutrients externally through hyphae and do not use phagocytosis. They have cell walls. Option D, Plantae, are multicellular photosynthetic autotrophs with cell walls. The organism described is clearly a protist, likely an amoeba. Protista is a diverse kingdom including protozoa, algae, and slime molds, characterized by being eukaryotic and not fitting into other kingdoms. The presence of pseudopodia and phagocytosis narrows it to protozoan protists. Question 4. A biology teacher is planning a laboratory investigation for students to explore the factors that affect enzyme activity. The teacher provides students with catalase enzyme extracted from potato tissue, hydrogen peroxide substrate, and various buffers at different pH levels. Students are instructed to measure the rate of reaction by observing the height of oxygen bubbles produced when the enzyme and substrate are mixed. One group of students hypothesizes that catalase will work best at a neutral pH of 7.0 because that is the pH of potato cells. They set up 3 test tubes with pH 4 buffer, pH 7 buffer, and pH 10 buffer, add equal amounts of catalase and hydrogen peroxide to each, and measure bubble height after 30 seconds. Their results show the highest bubble height at pH 7, moderate height at pH 4, and very low height at pH 10. Which of the following conclusions is most strongly supported by their experimental data and biological principles? Is it? Option A. The data conclusively prove that catalase is completely denatured at pH 10 and cannot function at all under alkaline conditions, which is consistent with all enzymes having optimal pH near neutrality. B. The results support the hypothesis that catalase has optimal activity near neutral pH, but the moderate activity at pH 4 suggests the enzyme retains some function, which is consistent with the fact that enzymes have a range of pH tolerance before complete denaturation occurs. C. The experiment is invalid because measuring bubble height is not a quantitative method, and the student should have used a gas pressure sensor to obtain reliable data about enzyme activity rates. Or D. The moderate activity at pH 4 indicates that the pH 4 buffer was contaminated with some neutral pH solution. Because enzymes cannot function at all at pH 4, due to complete disruption of hydrogen bonds in the protein structure. The correct answer is B. This question requires understanding of enzyme structure and function, particularly the relationship between pH and enzyme activity. Enzymes have optimal pH ranges where their three-dimensional structure is maintained and active site configuration is ideal for substrate binding. However, enzymes typically show a bell-shaped curve of activity across pH ranges, with some activity above and below the optimum before complete denaturation occurs at extreme pH values. Catalase from potato tissue does have an optimal pH near neutrality, but it retains some activity at moderately acidic pH as the data show. Option A is incorrect because it overstates the conclusion. The data show some activity at pH 10, and enzymes can function at various pH values depending on their structure. Option C is incorrect because bubble height, while less precise than electronic measurement, can provide valid comparative data for a classroom investigation. Option D is incorrect because many enzymes do function at pH 4. Pepsin in the stomach is optimized for acidic pH, and while catalase is not, it may retain partial activity. The most scientifically accurate conclusion is that the data support the hypothesis while acknowledging that enzymes have ranges of activity. Question 5. A student is examining a cross-section of a leaf under a microscope and observes numerous small openings on the lower epidermis, each flanked by two specialized cells. The student correctly identifies these as stomata with guard cells. The teacher asks the student to explain how these structures contribute to both photosynthesis and water balance in the plant. Which of the following explanations accurately integrates the functions of stomata with plant physiology? Is it? Option A. The guard cells contain chloroplasts and perform photosynthesis to produce glucose that is transported to other plant parts, while the stomatal opening allows oxygen needed for cellular respiration to enter the leaf. B. The stomata open when guard cells become turgid due to potassium ion uptake, allowing carbon dioxide to enter for photosynthesis, but this opening also results in water vapor loss through transpiration, creating a trade-off between gas exchange and water conservation. C. The stomata remain open continuously during daylight hours to maximize carbon dioxide uptake for photosynthesis, and any water loss is insignificant because the plant continuously absorbs water through its roots to compensate. Or D. The guard cells respond to light intensity by closing the stomata during peak sunlight hours to prevent excessive water loss, then opening them at night when carbon dioxide can be absorbed without water loss. The correct answer is B. This question tests understanding of stomatal function and the physiological trade-offs in plant adaptation. Stomata are critical structures that balance the plant's need for carbon dioxide uptake for photosynthesis against the inevitable water loss through transpiration. The correct explanation integrates the mechanism of stomatal opening, guard cell turgor changes driven by ion uptake, the benefit, CO2 entry for photosynthesis, and the cost, water vapor loss. Option A is partially correct about guard cells containing chloroplasts, but their photosynthesis primarily provides energy for guard cell function, not for export, and stomata do not primarily function for oxygen entry. Option C is incorrect because stomata do not remain continuously open. They respond to environmental conditions, and water loss can be significant enough to cause wilting if not regulated. Option D is incorrect because most plants open stomata during the day when light is available for photosynthesis and close them at night. CM plants are the exception with nighttime opening. Option B correctly describes the mechanism, the benefit, and the trade-off. How many did you score out of 5? Let us know in the comments below. Also, tell us which exam we should cover next. If you found this video helpful, please like, share, and subscribe for more.